Welcome to Beyond Shodh, a podcast celebrating the research, innovations, and startups in the field of science, technology, engineering, and mathematics. With every episode, your host Dr. Sonia Balian will bring you an inspirational and exciting STEM story through the eyes of experts, scientists, innovators, and researchers. Today, I am very excited to begin this journey with a series called Beyond Her Shodh, celebrating the contribution of women in STEM and beyond. Today, we have a very special guest with us, a well-recognized science enthusiast, a passionate and energetic scientist, and an excellent mentor, Professor B.K. Thelma with us. Professor Thelma is superannuated from the Department of Genetics at the University of Delhi South Campus. Currently, she is acting as National Science Chair in the same department. She has immensely contributed in the field of human genetics and medical genomics with translational importance for societal relevance. She has many prestigious awards in her name and published over 100 articles in international journals with several patents. So without a delay, I welcome Professor Thelma to Beyond Shodh. Thank you, ma'am, for accepting our invitation and welcome to our show.
Thank you very much, Sonia. I'm very grateful to you for taking time off to do something like this, and which I hope I will do justice to.
So first of all, ma'am, tell us about your vision and the research area of your laboratory that you are pursuing, and also how you have got started in when you joined this department.
Okay. Broadly, I would say the vision that I have is, let's say, genetic well-being. If I said so, that's health and well-being, but from genetics point of view, because we are talking about a large number of genetic disorders, and I really hope that in today's context, what we talk about as P4 medicine, that is predictive, preventive, personalized, and participatory, will actually be a reality. So that's, it's been the vision all through. As far as the focus is concerned, as I said, I'm entirely looking at only those disorders which have a genetic basis. If I have to just explain just a little bit, when we say genetic disorders, it can be a single gene disorder which is running in a family from a parent to a child, or both parents contribute to the child, and so on. And it's only about 6 to 8%, but a large majority of disorders are lifestyle disorders, about 60%, which includes things like hypertension, diabetes, asthma, rheumatoid arthritis, and so on. So we know very well that there is a genetic component even in these disorders, but there is a well-documented non-genetic factor. That is, it could be anything, we just say non-genetic factor, which is what makes predictions difficult in that group of disorders. So I've had an opportunity through my career to look at both the genetic basis of single gene disorders as well as try and uncover as much as we can for the genetic basis of the so-called complex traits or lifestyle disorders. So that really has been the theme of work ever since I started human genetics work, maybe in 1989, 90 onwards.
Can you just revisit your childhood and all those insights from your family values and your upbringing, and how they have transformed a little girl from cook to a top leadership position in human genetics?
Okay. At the outset, I would say that the family support, the family values that they instilled were very, very immense, and I think really the most important in shaping up what one grows up to be. So while there was abundant love, there was equal amount of discipline, which was very essential as a part of the bringing. So those are like really very important. And somewhere the value of education and to be independent, to have a career, was equally instilled more by, I would say, by practice rather than by through preaching, because the family was educated. My maternal grandfather was a teacher, but my mother had only sisters, but all of them were educated. And in fact, four of them, including my mother, were garment servants. The first aunt of mine being the first garment servant to be in Cook district under the British government. Okay, so that having been the background from which I came, a middle class family, so education with a purpose was really what we were kind of made to realize very early on was the most important, and that I think was
the driving force
to get where one is now.
You being the role model and inspiration for our generation of scientists and also the budding researchers. So, is there any role model that you look up to, or any inspiration that you look up to?
We just said that I was from a small town in Cook, so we really didn't have so much of exposure. Okay, neither by way of lots of books or those days, of course, TV and other kinds of media was not there. So I don't really recall reading or listening to anybody who kind of made that impression in a big way. But I think all through, even during, so when I stepped out of school, that continued to be the same. College, again, was not very, very influential in terms of a person or a few people. But the driving force, of course, was to achieve something. You needed to follow a passion, or needed to follow a path that you have chosen to be in, and so on. But I must say that when I got into my PhD mentor, Professor S.R.V. Rao, was a large source of inspiration because he was a wonderful human being, a great teacher, a very passionate scientist who really instilled values of what you do is not important, how you do is important in science. Basically, these are all very essential for the practice of science.
Yes,
so I think I learned a lot from him, and I've tried to follow that through my career.
Okay. Your areas of research is Genomics. So is it the integration of Ayurveda with the modern genomics, and how you got interested in this kind of interdisciplinary area?
Okay, thank you for asking that question because that's something very close to my heart. And I already said in my previous few sentences that the lab is focused on complex traits as well, uncovering the genetics of complex traits. So 20 odd years of work on complex disorders like schizophrenia, Parkinson's, rheumatoid arthritis, and inflammatory bowel disorders, and looking at global literature. We just realized that no matter what the technology has been over the years, we don't have any answers to the actual genetic determinants of complex traits, which means prediction and prevention, which are the key to genetic disorders, and addressing them in a translational way is simply not possible for complex traits. So, since we're talking about technology advancements, we started off with the recombinant DNA techniques, and then we started looking at candidate genes, and then the technology moved forward, and they said, okay, we now have whole genome sequencing information, and therefore we can do genome-wide studies. And from genome-wide studies, they had huge case control comparisons, hoping that they would be identifying the determinants of the disease. Didn't give us any answers. Then we said the routine whole exome sequencing is not good enough. Everything should be whole genome sequencing. We still don't have answers. And now, of course, everybody thinks about artificial intelligence and machine learning as probably some kind of tools which might just give you some answers. But I'm very convinced that what we need to address is really the heterogeneity, both in terms of clinical heterogeneity and genetic heterogeneity, which is a part and parcel of all these complex disorders. Okay,
Sure.
And there is no way as of today with the modern medicine and all of the tools in their kitty to address this issue. Okay. On the other hand, Ayurveda, the ancient wisdom, okay, and the practicing, the medicine which was practiced earlier, which is continuing to be practiced now, has a very different way of looking at health and illness. So the basic difference is, when you talk about allopathic medicine, you're talking about treatment of a disease, which is symptomatic. Whereas in the case of Ayurveda, the whole philosophy is maintenance of health and prevention of disease. So there's a lot of emphasis
And without
which is
side
given.
effects.
Yes,
Without
and which is, that is what is given the emphasis. And if they talk about maintenance of health and prevention of diseases, that means the principles of Ayurveda already were very different from the symptomatic based modern medicine that you practice. So, this is something which we have known, but unfortunately scientifically not validated sufficiently in the case of Ayurveda. But while we were stuck all the time with progress in complex trait genetics and to kind of make anything predictable or testable in terms of using it in a clinical setup, I realized that Ayurveda, with its deep phenotyping principles, which allow Ayurveda Vaid to put people into different categories. Each individual is unique. Genetics also says each individual is unique, but they didn't use the word genetics. They didn't talk about genome components which are coming from the two parents contributing to a child which is unique, except in monozygotic twins. But they call each individual as unique based on the prakriti or the constitution. So their way of assessing an individual allows them to group people into broadly three categories of Vata, Pitta and the Kapha. That's the predominant groups. And then you have a larger majority who have the mix. So you can have Vata, Pitta, Pitta, Kapha, Vata and so on combinations. But if you are able to even pick up that small group which is fitting into the Vata predominant or Pitta predominant or Kapha predominant, we believed very many years ago, maybe close to 15 years ago, we actually thought we are quite, probably we will get a lot more insights if we used the Ayurveda principles of deep phenotyping, get individuals put into homogeneous subgroups, and then use all of the currently available technologies to answer the questions on what really underlies what are the genetic determinants of these diseases. So with the limitation that we had with the contemporary medicine and the way that the limitation that we could have, we had no way to address the heterogeneity in any disease, doesn't matter whether it's type 2 diabetes or rheumatoid arthritis or hypertension. The deep phenotyping principle was what I thought was going to give us really some deep insights. So those were the early years. Luckily, we could just kind of we had we still have the excellent collaborator in Dr. Bhima Bhatt, who was the Ayurveda specialist. He worked at Holy Family Hospital for many years, a product of Jamnagar, one of the best Ayurveda schools in the country. And currently he's at Medanta, having opened a department of Ayurveda for this kind of a complementary medicine.
Did you also practice yoga and Ayurvedic techniques in your daily life?
I'm a regular practitioner of yoga, and I learned yoga, with from many different schools, but currently I follow the Art of Living set of the pranayama, the yoga of course, and then followed by pranayama, sudarshan kriya, which I think is extremely important for one's well-being and for one's positivity. So there was all these combinations kind of help, but then the Ayurveda principles really, I think, have the answers, will give you the answers to the limitations which are currently faced by complex trait genome researchers. So that's really, I think, the thing. That's how we started off with Ayurveda traits.
So you got the good result. I mean,
So we were talking about Dr. Bhatt. So we decided that we will write. He was very convinced that Ayurveda has some of the best ways to address complex traits because they treat each individual as unique. They have recommendations for food, they have recommendations for lifestyle, because they know that each prakriti comes with its own baggage of certain diseases. So if you follow their guidelines, they're able to kind of then, at least you will be able to maintain health. And if by chance you have an illness, of course there are ways to set that balance of elements right, because it's all, we're all a combination of 5 different elements. Somebody will have one element more and less. That's a combination, right? So he was very convinced that we will have answers. And so we wrote a project to the Ministry of Ayush, and we did candidate genes and looked at the Vata, Pitta and the Kapha groups, and he suggested that we don't touch diabetes, although it's the most common condition, because in Ayurveda, the diabetes, type 2 diabetes, is not called as just one category. There are 20 different subtypes. So you can imagine what happens in a diabetology clinic in a modern medical center versus the way Ayurveda wide actually treats. So he said if we require a minimum number of samples, diabetes may not be the best, because within a 3 year span, we may not get the required number under each of these predominant Prakriti groups. So he suggested Ayurveda clinics see a lot of rheumatoid arthritis patients, because people after a certain period really get debilitated with rheumatoid arthritis, because either they stop responding to the drugs or they're not really taking care. And they all end up going, or most end up going to the Ayurveda clinic for the Panchakarma treatment and things like that. So he said there's a very rich flow of patients of rheumatoid arthritis. So that's how we wrote the project. We got that. We looked at candidate genes, and the results were fascinating. And we published, I think the first paper which we published was in 2012, where we actually showed that there were differences in the genetic susceptibilities between the three groups. And then, of course, now in between, I had lots of other modern things which were happening with all my other diseases. But my National Science Chair project is exclusively focused on our genomics, but not just with genomics, but we have an integrated omics approach where we are looking at genomics, gut microbiome, then we look at urinary metabolome and proteome. We look at epigenome, and then of course the phenome is there. And then we think that if at all this data set is too large, we might also use the AI ML methods to address. So this is really the project which I'm passionately pursuing, and I'm keeping my fingers crossed that we will have some nice insights.
Yes, definitely. Yeah. So ma'am, you are an advocate in the area where you study that newborn screening and P4 medicine, as you have already described, for early detection in inborn errors through your research. All the tests that we have currently, is they are adequate, or we require some more research, make some tests mandatory so that we can...
Perfect. Okay, you seem to be asking all the questions which are really, really close to my heart and which I'm very passionate about. Again, ever since I started with human genetics, we're all students of genetics, right? So we have known Sir Archibald Garrett's inborn errors of metabolism. And that's something where after a child is born, based on the metabolite profiling, you can actually say what the genetic defect is, and mostly they are autosomal recessive disorders. Okay? And for years, since I started my human genetics research, I've been actually trying to get support to go ahead and do a newborn screening between different agencies, whether it is DBT or ICMR and so on. But it just didn't happen. There's a lot of discussion, a lot of talk, but it really didn't get off. So to answer your last question first, in our country, we don't even have newborn screening as mandatory. Okay? It's over 60 years since in the West, it's routine. Okay? Depending on the epidemiological data that they have, they know which conditions need to be tested on a mandatory mode in the newborn. But unfortunately, till today, newborn screening is not mandatory. A very, very, sharp nephrologist will probably say, okay, look, this child seems to have this condition, and therefore, under suspicion, or diagnosis, they will go do a testing. But how many are able to get the test done? How many are able to start the intervention in time is the most important. So for newborn screening, once you have diagnosed the child is positive for the IEM, for some of the IEMs, you have treatment possible. And if you have the treatment within a certain period of time, the disease will not manifest, or will be in a very, very milder form. It will not be debilitating, or it will not be a lethal condition. So this is something which we really thought was a very doable translational science, but didn't get support. But luckily, when I happened to be a member on the Scientific Advisory Council to the Prime Minister of India during 2009 and 2014, in the year of science, it was a year of science early on 2000. One of those years was that, and all the members were asked to kind of propose, what would you like to do? So I said, I really think we should do something science for society. So the ball was back in my court, and they said, come with a proposal. So I said, okay, I have a very passionate proposal on newborn screening. I'll come back. So I made a presentation to the committee on newborn screening. So I had to tell them what the disease incidence is, what is the disease burden, and how preventable some forms of them are, how treatable some other forms are, and to show them the lacuna in our country. And then for several months, the talk went on between different funding agencies, and nothing really happened. And one follow-up, the credit entirely goes to Professor S. Ramasamy of the Department of Science and Technology, okay, who said, Thelma, come back and I will support this project as a feasibility study. Okay, and again, since then there was no looking back. He had wanted us to show the feasibility in a place like Delhi as well as in a slightly rural setting. So we had decided, okay, we'll do one in Bihar and one in Delhi. But when we finally made the project, he just said, I think we should not kind of go and take it up in Bihar yet. Maybe we should show the feasibility in Delhi. And then we put together with my clinical collaborator, Professor Seema Kapoor and Maulana Azad, and 20 other hospitals across Delhi. We put together this project and we screened 200,000 babies in a matter of about 20 months. Okay, and we tested them for 5 treatable conditions, and we generated epidemiological data for remaining about 31 conditions. Okay, because ethically they would not allow us to do anything beyond for those conditions where there was no treatment available. So we were very fortunate that we could do testing and treating for 5 conditions and generate the epidemiological data. So we have generated the epidemiological data for all of the then testable IEMs, inborn errors of metabolism, for the first time. And based on that, the Delhi government has already made this program called Mission NEEV, okay, NEEV, Neonatal Early Vision program, which is on at Maulana Azad Medical College now, where 2 conditions, congenital adrenal hyperplasia and congenital hyperthyroidism, together with G6PD, is being routinely screened for newborns coming from all the government hospital, newborns coming from the government hospitals. But my passion is to kind of really get this, because I view this as a data to policy research, and my sincere efforts are to make this into a policy, to make it mandatory screening in the country. I hope that dream of mine will come true, but for that I know that we need to have a little bit more epidemiological data, because India is vast, very diverse. So we really need to have, at least for a few representative states, we must have the data before we can say which should be the conditions which are made mandatory. Okay, that's very, very important. So this is why, and that's a classical example of A, translational medicine or a technology for the common people. B, it's a very, very early example of before medicine. Okay, it is predictive, preventive, and participatory, of course, and personalized. Okay, so wherever there's and these days with so much focus on rare disorders, okay, more and more emphasis is on developing newer therapeutic molecules, cost-effective or affordable therapeutics, and so on. So while a large majority of the public is still unaware of these conditions, okay, we have a chance. If parents are unaware and they get a first baby who's already affected, which is screened in the newborn, we can't do anything for that baby, there's no treatment. But because we do the genetic analysis in the parents and identify the mutation, because we now have the child who is affected and the parents, for the next pregnancy, when they plan the family, we can actually do predictive testing for them. And so I think, it's truly a P4 medicine example, and something which is so doable and so inexpensive. It's not that it's not, it is inexpensive if you really do the test, because all that you require is a drop of blood from the heel of a baby, newborn, 24 to 48 hours after. That is after the child had its first feed and the own metabolite profiling can be done. We just need to do that, and it's very, very inexpensive to cover all of these tests. And of course, if there's early history of another baby born early in the family, then you know that genetic testing is possible, prenatal diagnosis, and it can be very easily prevented. A defective baby or a baby with a genetic disease condition can actually be prevented from being into the world.
What exactly is one milestone, or you can say the turning point that shaped the focus of your laboratory?
Okay. I told you that I wanted to be a medical doctor, but I started off working with a rat, a very special mole rat, which had what was called as fragile sites on the X chromosome. And we know that fragile sites on the X chromosome are responsible for, or are an indication of, Fragile X syndrome, the most common form of inherited mental retardation in humans. Okay, so while we were working on the Fragile X chromosome in the animal, we did lots of different, we could kind of do some fancy things, including micro dissection of the regions and cloning and trying to, characterize. But there wasn't really a great breakthrough because you're just working on one odd rodent species. But 1991, so that was just exactly 4 years after I joined the Department of Genetics, while I was thinking how to get back to my passion of doing human genetics, the gene for Fragile X syndrome was identified by Ben Oostra and his group in the Netherlands, and we were lucky to get that probe. And Professor I.C. Verma at All India Institute of Medical Sciences was also interested because they had an active genetics diagnostic unit there. But I think we went ahead and we could just kind of do a much larger study on molecular diagnosis for Fragile X syndrome, because our approach was to go to the schools which are housing these, which cater to these special children, and with Professor Meena Gupta, who was a clinical collaborator from Neurology GB Pant then. Every Saturday, we would just go and meet parents, we did the parent-teacher meetings and so on, and try to explain to them that some of these special children could actually fall into this category of Fragile X. Okay? And we didn't have any pictures of a Fragile X positive child from the country at that point.
You have that list of symptoms.
Yeah, we just had the phenotypic symptom description. But all the pictures in books were all Caucasian kids, okay? But ma'am, Professor Meena Gupta tried to kind of tell them, look, these are the features. Have you seen these in your kids? Or can we actually look at your kids who are here? To kind of motivated them. And then parents said, yeah, my child has these symptoms. My family, in my family, a cousin has another similar child like this and so on. So that was really the beginning. And when we got the probe and we picked up the first child from the special school at Lady Evan College, they used to have this center for the special children. And it turned out that when we developed a southern blot, we actually saw that the probe identified the child who was positive, the mother who was a carrier, and then of course there was another child in the family who was a normal one. Father, of course, would only be a control. So we got that beautiful classical pattern that is expected for an X-linked condition, and one which is with a trinucleotide repeat expansion. So that, if I were to say, was really 20 years down the line of wanting to do medical genetics and biomedical genomics.
So when this incident happened, or this discovery happened?
It was by 91 the gene was identified. I think we already had, by 92, 93, we already had very many positive cases, because the minute clinicians realized that there was this test which is possible, we started getting samples from across the country. Several individuals who said there's family history and the symptoms fit into the Fragile X phenotype, they would send us samples. And so we have a large bank of Fragile X positive, or those who came for testing. So that was the time. And it was absolutely thrilling to see on a southern blot. Those days, southern blot was magic, right? So we just got that. And then there was, then I moved. So that was a neurodevelopmental disorder. And I told you my mentor, Professor Rao, was very, very good. Okay, he had a very little bit. So he always said, don't put all your eggs in one basket. He always told us that. Okay, meaning to say you need to have kind of a little parallel things happening. So we said, okay, while we are looking at neurodevelopmental disorders, they wanted a counselor to be as a part of this project and so on. So I happened to meet Professor Smita Deshpande, who was a psychiatrist, and who were desperately looking for a geneticist who would look at schizophrenia. So we went into neuropsychiatric disorder as another branch out. And then from there, we went to neurodegenerative, which is Parkinson's. So we now look at all the three conditions: intellectual disability, Parkinson's disease, as well as schizophrenia, the three main parts of the broad neurological conditions, but everything from a genetic basis. Okay, so we were lucky; we got families with these conditions, because if you just take sporadic cases, the chances of you identifying good leads, genetic leads, are very little. But we got nice families, and therefore our Discovery Genomics went in a big way. 2014, we already identified a new disease-causal gene called MID2 for inherited intellectual disability, again X-linked, and now it's a part of the panel, global panel for testing for intellectual disability. We already, and of course many more. We have had Parkinson's again. For families, we've identified new disease-causal genes which are also there in the OMIN, which is on Mendelian inheritance in man. We have these numbers which are given for discoveries. So that's there. And then of course schizophrenia, we had really large families, and we got very useful or very good insights into the oligogenic basis of schizophrenia. So although there are families, it's not one gene. It is a set of genes.
Extensive work and hard work of so many people.
Yes.
Collaboration, hard work.
So my largest, my first and foremost gratitude is to the clinical collaborators, because without them, I could have just not touched a patient or never got access to anything. Yes. So starting from Professor Meena Gupta, all through my other collaborators with Dr. Madhulika Kabra at AIMS, Dr. Smita Deshpande at RML, Professor Madhuri Bihari and Professor Uday Muttani on Parkinson's, from Bangalore, from AIMS and Bangalore respectively, and these are only for the neurological or the brain disorders. Okay, I'm so very grateful to them, but for them, we could never have done biomedical genetics in a university setup. So funding agency is something else. DBT, I need to really thank them for that, because right from Fragile X project, which was granted, the first one to be granted, I've had continuous funding support up until, it's continuing to be there even now, and there is no way you can thank the clinical collaborators and the funding agencies, and of course the university for allowing you to do, because I'm committed to teaching. I need to be my 200% for teaching. But beyond of that, university allows you to do what you want to. You can pursue whatever area of research. So I think the freedom, the autonomy, and the support, whatever that we got, is hugely valuable.
That you have trained a task force of geneticists, and which again currently working in different domains and with different special positions and senior positions. So what were the few mantras that you always pass to every student, that this is the lead, or you can say this is the mantra that you have to keep with you while you are pursuing your research career? And
of course I said a few things, but as I said early on, like the family, nobody tells you in words, right? You observe what your parents are doing, what your grandparents or the extended family is doing, so that if you're sensitive, you will imbibe those values. Yes. And I believed that you don't have to preach, okay? You need to kind of, they have to pick up what you are practicing, or rather you should actually be a role model, because they should realize, okay, there is no substitute to sincere hard work, okay? That's something which is primary. You can be brilliant, but if you don't have those basic things of passion, which is essential, and then dedication, your hard work and sincerity, you will not make it anywhere. But so I guess I had really wonderful set of students, and largely for the longest period I had a lab only full of probably girl students, and probably there was one odd boy there, and which only in the last 2 years, in the last few years, I actually got more boys and less of girls. Otherwise, I think they all saw that, if I'm the first to come to the lab and if I'm the last to go to the lab, the message is very clear, okay? And for me, it was, I can take the horse to the water, I can't make it drink, right? So I think somewhere along, they got the support from me, sure, but that was my duty. If I took them as PhD students, it was my moral duty to make sure I guide them, mentor them, and point out when things are not the way they should be. But rest is really up to them. And they're all doing very well. I have the last 2 to finish their PhD now, and only sad thing which I have, a regret that I have is, I can't say it personally, it's a regret for me, but for them it's very good. Except for 5 students of mine, everybody is abroad, and they all are in the industry holding really good positions. Okay, and even the ones who chose to stay back.
In India also, we need good scientists.
I know, but
so if you are not in India, then what will happen?
No. So that's no. So I must tell you that when I went as a postdoc to Switzerland, I was in Basel at the Children's Hospital, because that was my beginnings of my first steps in human genetics and human medical genomics. So they offered me a job. I said no, I will not stay in Switzerland. I will go back to the country. So I came back to India without a job.
Good
Luckily, 6 months after, I got what was called as a pool officer. CSI used to have these pool officers. I worked as a pool officer, then I worked as a research associate of the university, and luckily for me, genetics department had an opening, and I made it to the genetics department, which was really, I felt, the place I had to be. So that's what it is. So it's your choice. So now I have students who want to come back, but the respective spouses are not willing to come back. So all the training has largely been, been utilized, or the training has benefited the West more than our country. But I'm still hopeful that the ones who are here will have their own group, breed which is coming up, and they'll contribute.
So ma'am, we have a little surprise for you. I hope you like it. And
what is it?
We have some of your students.
Shruti and Sumedha dear,
oh my God!
We wanted to let you know that you have always inspired us to do the best of our potential, and we have been very sharp and committed to our work because of your influence, and which in turn has helped us turn into effective communicators and how to strategically maintain relationship with our peers and make an impact on the lives of the patients.
And I also want to especially thank you for having fostered such an amazing environment in your lab. Like, you have trained up a whole generation or two of scientists to have an incredible work ethic and a great sense of commitment to working together as scientists, working collaboratively and working globally. And as a result of this, many of us have ended up in very successful positions. And I'm also very thankful to you for having fostered a great sense of community in your lab of students present and past, which is why, like, Shruti and I are now together at the same company as well. So I wish you all the very best, ma'am, and thank you again for being such an amazing mentor and someone we really look up to. And we wish you all the best. Thank you.
All the best, ma'am. Thank you.
Dr. Thelma is a well-known scientist. But today I would like to talk about her as an educator and a mentor. She held all her students to very high academic standards, and personally, that helped me to learn perseverance and resilience in a professional space. She emphasized on getting the basics right, getting a strong foundation of concepts, and that helped me a lot. One of my current jobs programs, and I find myself emphasizing pretty much the same things to my students as well.
Hello everybody.
You really kept this as a secret. Oh my God, this is too much.
I'm very fortunate. I feel I'm very fortunate that I got an opportunity to get to know her since my master's time as teacher, and then subsequently as mentor and supervisor during my PhD time. It's been more than 1.5 decades that I know her. Till now, I get many valuable inputs, suggestions, even sometimes, you can say handheld, guidance from her. She's been very fantastic teacher and supervisor for all of us. She's one of those, scientists or academician who believes in preaching what she practice. She firmly believes in importance of communication in science and management skill. While doing science, we all know that she pushes her students for scientific communication in a language that understood by all. I don't remember any time where she used to impose her ideas on students. Basically, while doing PhD, we used to nurture our own interest, own ideas. When she used to support us wholeheartedly. I don't remember any time where we were left alone. No matter how busy she was during that time, she always gets very, very dedicated time for her students. Starting from study design, result interpretation, experimentation, writing manuscript, communication. Time for our students. She used to guide us from scratch till the publication comes online. So that's the kind of dedication basically, Ethics also is very important to stick to the ethical guidelines.
That is important.
So she used to teach her PhD students how to follow those guidelines very strictly. Management of samples, she used to tell us that, look, every samples are important.
So her support and guidance actually have helped me a lot during my research time, and I'll be always indebted to her. And among many students of her, currently I had the privilege to be her student during my MSc course in genetics in 2014 to 16. And the way that she has taught me the human genetics and other students, those concepts, basic concepts, are still etched in my mind. And the way that she interacts with the students, as you know, she's an excellent teacher and ask students all the questions and try to get the answers from them. The way that she teaches, it's totally different and a very unique way, and we learn a lot of today of that today also, and it's currently being practiced in my research too. So not only in teaching, but she is also a great mentor, a supervisor, and which actually has helped me, and she has actually helped me a lot for my research work. And basically, I was a student, PhD student, and she actually has given me the confidence to work on stem cells and CRISPR-Cas9, and also how to write papers, review manuscripts, even write letters, participate in various conferences, present our work, presentations, how to put all the slides in places, and all those things, even for projects related work. So she has given me a lot of confidence, and I'm very grateful to be her student actually. And at this point, I also would like to say that I actually wish her very good health after retirement. And as always, she says that you should always give 200% and not 100%. So those things actually helps you to actually take your study as well as your work life forward. And also besides academics, she has been a great inspiration person for me. And even having all those achievements, being down to earth, and not for only for me, but for other students also. So I actually wish her very well for her health and have a great life.
I mean, bottom line is you have to be passionate, in what you're doing. And as I said, as Nandisha was saying, give your 200%, because that's really what I say. There is no compromise.
As the representation of women in the top leadership roles bothers you on that? Because in the medium level, while doing MSc and PhD, the representation is quite well.
Yes.
But
As you
go higher, the representation is somehow decreases.
I've said this before, and I will actually repeat it. For a woman, the age between 30 to 35 years is extremely critical.
Yes.
If we only had a support system which would give them that allowance or that support during those 5 years, women can reach to greater heights. Okay, because invariably a woman, you will agree definitely, and I'm sure the men folk will also agree, no matter what, a woman has a lot more responsibilities than a man does. Okay,
and it comes naturally.
Yeah, it comes very naturally, because these are like, you can say that's biology. Okay, but given that you have a job, you have a home to look after, you have kids to tend to, and so on and so forth, and a lot of other responsibilities, you have to spend so much more time beyond the regular working hours if you have to make it. Otherwise, most women say, okay, no, I have a job, I'm happy doing a 9 to 5. So while they got a PhD and they might have done brilliantly, they just don't give that same kind of a commitment for a subsequent pursuit. Those who have been fortunate, and I will say, I've had the luck, I've of course had the determination, the family support, which is very, very important for a woman, without which she can't take one step out.
Agreed.
Okay, so I've been lucky on all those fronts. But given that, woman, I think there is no reason. I like to say it for the nth time. I always tell them that don't tell the world about the labor pains. Show them the baby.
Yes.
Okay, nobody is bothered about your troubles and your problems.
Yes, sure.
Okay, they all want to see at the end of the day, are you putting something comparable to another scientist, your colleague? That's all that they want to know. So while there should be no difference between male and female and so on and so forth, a woman, given her responsibilities, multiple responsibilities that she kind of has to undertake, I think a little support system in the early years would go a long way. Because if she says that, okay, once my child comes to class 10 or class 12, then I'll get back. Time is gone. Science doesn't wait for anybody. Ideas are nobody's monopoly. Things have gone on, and then you'll feel redundant. You'll say, okay, look, I'm so behind, I can't do. And how many of that? How many people have that passion to pursue despite all odds? So these are difficulties which a woman faces, and which I think. But now, having recognized this, the government has, the various funding agencies have come up with new different programs exclusively for women, relaxation of age, etc., etc. But to answer your question on whether they really have the representation top, the answer is no, not really the way it should be. There are several times that I'm the only woman on some on some of the committees or whatever. Yes. But it's unfortunate. Okay, we could have a lot more representation, because I think women, despite all their responsibilities, use their time very judiciously and give their best. So while we don't lobby, okay, it would be very important that they get that due recognition without any kind of a bias, because hiring still is very biased, because they have that thing at the back of the mind.
Yes.
Oh, lady candidate, she will go on maternity leave, so there will
Yes.
be all these things are still existing.
Yes,
Okay,
they still
unfortunately.
exist. Yes.
Unfortunately. So I wish these things will change. We should have changed by now, considering we are otherwise so progressive. So that's where I always feel, the progressive thinking of my family and the support, I think really, was huge. And that's so true for all of the girl students. I told you, at
Yes.
one point I had only girl students in my lab. And I really wanted them because it was just nice. It was just nice that you could really train a generation of them so that they could take forward. If you have the passion, you will find the way. And
Yes.
if you have the will, you will,
Find the
if you have the will, you will find the way, and your systems will fall in place. Okay? Because you can be troubled a little bit, but so you have to learn to say that, okay, the secret of success is not really wishing for things the way you want them to be, but to actually work with things the way they are. If you have that acceptance, it's a flow. It's an easy flow. So that's my message, 200%. Give your 200% and don't wait for your, don't expect results. You do your best and leave the rest. Okay? That's.
So, ma'am, at the end, how do you see your journey? Are you satisfied? Or you still get the thirst? No, I have to do this, I have to do that, and still several things that I.
I must say that most of what I had planned, or most of what I wanted to achieve, of course, with science, one goal leads to another, because each of my projects have just opened up. One project you finish, it opens 100 questions. So I pursue the next and so on. So as I said, P4 medicine is very important to me. So the way I look at it is things like getting science, the fruits of science, or fruits of all this so-called genetic medicine and things like that for the societal benefit is one thing for certain. The second thing is genomics, because that's where I think we can contribute to global science. Here we are talking about, newborn screening is what we're talking about, health for all in the country, in a developing country. But when you talk about things like genomics and the fallout which may be there, which I'm very hopeful will be very good, is going to be for global benefit. So I think, there is never a time to never end. So while I know my limitations, that I am retired, I have a few years in this position as a National Science Chair, I hope to be able to contribute to a science and for well-being in one or the other way. That I'm very confident, God willing, that you have the good health, okay, and your other faculties are all in place. So
we wish you all the good health, and thank you so much for sparing so much time actually from your busy schedule and sharing your valuable thoughts and experiences with us. It's memorable for me, and thank you so much, and we are very grateful to you for this gesture.
Okay, I must thank you for kind of, giving me an opportunity to share my thoughts, which I think from time to time is important, because I know I have a bunch of students who have seen me work 24/7, but then for the larger majority of women students who might have concerns, any small message which might go out of this is a huge thing. So thank you for making that happen, or for enabling such a thing to happen. Thank you very much.
Thank you so much.
Thank you, and good luck in your own efforts and in your own career. I have been very fortunate to have highly motivated, pleasant, and a productive set of PhD students all through. All 5 fingers, you will agree, are not the same, but each one of them had their own strengths and were just lovely as individuals, which contributed to the health of the lab. While the broad research theme in the lab remained discovery, functional genomics, some bit of translation, the technology changed over the decades, but each of the students were able to pick and run with those and make significant contemporarily relevant contributions, which is what the lab, I think, can claim as an achievement or sustenance in science. I'll be literally failing in my, sharing my thoughts. It would be very unfair if I don't add the most important part. That is, whilst PhD students came and went, my scientific and technical staff, especially Prachi, Anjali and Vinod, remained my lifeline through a large and important part of my entire research journey.